Hi, this is Alex from devstories.eu. AlphaFold Protein Structure Prediction, Episode 4 of 21. Training a breakthrough machine learning model is impressive, but for a working biologist, a raw neural network is just another heavy tool to configure. To truly change the field, you do not just publish the model. You use it to fold nearly every known protein on Earth, and you put the results online for free. This is the AlphaFold Protein Structure Database. Built in partnership between Google DeepMind and EMBL-EBI, this database holds over 200 million predicted structures. That massive number represents almost the entire known protein universe. Instead of setting up hardware and running the AlphaFold algorithm yourself, you bypass the compute entirely and query the completed predictions. Consider the workflow for using the web interface. If you are investigating the free fatty acid receptor 2, you go to alphafold.ebi.ac.uk and type that name into the search bar. You can search by gene name or sequence, but the most precise method is using a UniProt accession number. Because the database maps directly to UniProt, every structure is tightly linked to existing biological metadata. When you select your target from the results, you land on the specific entry page for that protein. This is the part that matters. The central feature of the entry page is an interactive 3D viewer, but it does not just show you the physical geometry, it visually represents the model's internal confidence in that geometry. AlphaFold scores its own accuracy for every single amino acid using a metric called PLDDT. The 3D viewer color codes the physical model based on these exact scores. Dark blue indicates very high confidence in the structure, light blue is confident, yellow means low confidence, and orange means very low confidence. When you rotate the free fatty acid receptor 2 in the browser, you'll see sharp, dark blue helical structures where the protein crosses the cell membrane, but the loose tails dangling off the ends might be bright orange. Those orange regions are rarely failures of the algorithm. They are usually intrinsically disordered regions, meaning parts of the protein that do not possess a fixed shape in biological reality. Viewing the structure in the browser is good for a quick sanity check, but actual computational work requires the raw data. Below the viewer, you will find the download section. You can download the 3D coordinates in the standard PDB format or the modern MMCIF format. You drop these files straight into your local modeling software to measure distances or simulate molecular interactions. Alongside the coordinate files, you must also download the predicted aligned error data. This is provided as a simple JSON file. While the colors in the 3D viewer tell you if a local piece of the protein is accurate, the PAE JSON file tells you if the relative positions of two different pieces are accurate. It contains a matrix of error margins for the distances between every pair of residues. If your protein has two solid blue domains separated by a flexible hinge, the JSON data will tell you if you can actually trust the angle between them. The AlphaFold Protein Structure Database shifts your workflow entirely. You no longer spend days trying to predict a structure. You spend minutes looking it up, and your actual work becomes interpreting the confidence metrics attached to the download. That's all for this one. Thanks for listening and keep building.